Hello and welcome to Invertebrate Paleontology and Paleobotany at Utah State University. This is lecture 9, where we'll try to answer the question, how can fossils reveal what the ancient marine environment was like? So this is the first of sort of 2 lectures where we'll be talking about paleoecology and how we can use the fossil record to sort of reconstruct what the environment was like. And in this lecture, we're really going to focus on the marine environment. So before we go into too much depth with reconstructing the marine environment using fossils, I wanted to kind of go through a couple terms, particularly in this lecture, to get us thinking about the marine environment and how animals and plants and organisms exist together in the fossil record and how they do that in the modern record as well, and sort of understand or detangle these complex relationships between various organisms. This is all part of the science of ecology. So ecology is the study of interrelationships between organisms and their environment. So understanding what organisms you have present in the fossil record will tell us a little bit about what the environment was like. And so that's the key of doing these types of studies. In ecology, you can go out to the actual environment and you can make observations, you can collect species, you can analyze, whatever you can see in the natural modern environment. With fossils, we are basically getting sort of the fragments of fossils that are preserved throughout a series of processes. So in this figure down here, we see that we start with the original community of organisms, of plants and animals. From that, we're dealing with only the ones that have died, their organic remains. From that, only those ones that have been fossilized, and of those, only the ones that have actually made it into a fossil collection. So paleoecological reconstructions rely on trying to sample as much of the original community as possible. Now what you want to do is reconstruct the ecosystem. The ecosystem is a portion of the environment and all of its organisms that it contains. And so to recreate the ecosystem of a particular rock unit, we're going to have to try to find as many fossils and diverse fossils that we can find in the rock record. Now there's a couple other terms I want to define. The first is ecological habitat. Habitat is basically the environment in which an organism lives in. This is like the address or house that the organism lives at. It's basically the type of environment that these organisms find themselves in. Habitat can be contrasted with ecological niche. The niche is basically the way in which the organism fulfills its duties, its way of life or role within the ecosystem. Niche comes from the term for niches, like you put into like a wall, like a little niche. But it's basically the job or the duties that the organism provides. So for example, with this clam here, it's a filter feeding organism that filters out various microorganisms within the mud or substrate, and that's its niche. Whereas there might be other niches that might be carnivorous animals. There might be other organisms that are herbivores, they're grazers. So those are all types of ecological niches. So that's the way in which the organism is able to function and survive in the environment. Another thing ecologists are really interested in, and less so in paleoecology, is figuring out food webs, or the way in which energy flows through an ecosystem. So you've probably been introduced to food webs in an introductory biology class or from high school or maybe even junior high, where food webs are basically how the energy, the nutrients in the ecosystem is transported between various organisms. So this is where you have herbivores, and the herbivores are eaten by carnivores, and the carnivores can be scavenged on other things. So it's basically following the energy through a complex series of relationships. Now food webs are really difficult to reconstruct using the fossil record, in part because oftentimes you're missing some of the key components of food webs, including many of the primary producers, which you may not necessarily see in the fossil record. Closely related to food pyramids are energy pyramids. Energy pyramids are where you take the total biomass. Now the biomass is basically measuring the total amount of living material, and then you basically categorize how far away they are from the primary producers. The primary producers are those organisms that derive their energy from sunlight or chemical reactions. So primary producers don't necessarily need to rely on other organisms to survive. So we have primary producers, primary consumers, secondary consumers, tertiary consumers. So as you move up the energy pyramid, at the tip are going to be organisms which rely on the previous, and they oftentimes will quickly narrow down to a very small amount of biomass. So think about this example where we have eagles at the top, snakes, squirrels, and peanuts. So there's going to be a lot more peanuts in terms of biomass compared to squirrels, and there's going to be a lot more squirrels in biomass compared to snakes, and there's going to be a lot more biomass of snakes compared to eagles. So the higher you are in these trophic levels in the energy pyramid, the smaller the amount of biomass. And trophic pyramids are usually not very high. Once you get about 4 or 5, you really just don't have enough energy. So reconstructing food webs can also help you a little bit in the terms of understanding how much energy is entering into the system. And this might be something interested in paleontology when we're comparing geographic places or trying to reconstruct sort of what the climate or the environment was like. So here's a comparison of the diversity of various organisms in a food web where we can classify phytoplankton and zooplankton in a faunal study. We're looking at the English Channel and Long Island Sound, and you can see there's greater diversity within the Long Island Sound than there is in the English Channel, indicating that there is probably more energy or more light hitting the Long Island than the English Channel, which tends to be a little bit cooler. And so understanding these sorts of food webs will give us a better insight of what the environment was like. And we can reconstruct this based on the fossil record and looking at these comparisons and reconstructing the food webs. Now, we live on land, and so many of us are probably not very familiar with what the marine ecosystem is. In the next few slides, I'll talk a little bit about the different terminology that we use for the different parts of the marine ecosystem. So this is a very simple diagram showing the transition from a coastline into the open ocean. And we can divide up this transitional zone into the coastline, the continental shelf, which is a shallow ocean that sits on top of the continent, the continental slope, which drops off from the continental shelf, the continental rise, and then the open ocean. Now, the continental shelf can vary in its width around continents or islands. In the Atlantic Ocean in North America, it's very wide, going for many kilometers, whereas in the Pacific Ocean here in North America, the continental shelf tends to be a narrower shelf going along the Pacific Ocean. So it tends to have to deal with the geology as well as changes in sea level over time. So many organisms are going to live in various parts of the ocean, whether up on the continental shelf or in the deep ocean. And these are very different environments, as we'll show. So oftentimes organisms, their habitat can be classified in 2 ways: benthic organisms and pelagic organisms. Benthic organisms are organisms that live on the ocean floor or the ocean bottom, whereas pelagic organisms live within the water column. Now, the benthic zone could include any organism that lives on the bottom, right, at the bottom of the ocean. So this could be along the continental shelf, or it could be deep within the deep ocean, the abyssal plain. Pelagic is the same way. The pelagic zone includes the Photic Zone up very shallow waters, as well as the deep, dark waters of the open ocean. So organisms, when we refer to them as benthic or pelagic, is really just basically describing where they live in the water column, with benthic being tied down to the ocean floor, whereas pelagic are moving around the open ocean or within that whole, the entirety of the water column. Probably one of the most important constraints on organisms is whether they live within the Photic Zone or not. The Photic Zone is that zone of the ocean which light penetrates down. Now, light can penetrate at different depths depending on how much or how many organisms or how cloudy the ocean is. So along coastal waters where you get a lot of the clay minerals that are dissolved load, you also get a lot of photosynthesis going on with organisms. So the water along coast tend to be a little bit more cloudy because of all the biological activity, as well as these clay minerals and things that are coming out from the continents. Light penetrates about 50 meters in depth, and that's not really that great thinking about how deep the oceans are. If you go out into the open ocean where you're not getting much sediment coming off of the continents, the light can penetrate down to about 200 meters. One of the reasons that the deep ocean waters are a darker, more rich blue is because it's the blue wavelengths of light that penetrate the deepest in the ocean. When you get to the coastal waters, they tend to be more greenish in color, and that is because the green light is the ones that penetrate the deepest in the coastal waters. And oftentimes, if you're flying in an airplane over the open ocean, you can see this contrast between the coastal waters, the green coastal waters, and the deep blue of the ocean, open ocean waters, and that has to do with how far the light is penetrating. Now, the photic zone is very important because many organisms that we'll see depend on that light as a very important source of energy. Along marine ecosystems, we can also sort of divide up the zones. So the coastal zone is often called the littoral zone. This is where usually you have the coast. A lot of times these are kind of called beach areas, but it also includes going off of the beach into the surf zone and a little bit further away from the surf zone. Often littoral zone is the zone in which wave base or waves coming in impact on the seafloor bottom. So it's a very high energy zone. Then we have the sublittoral zone. This is the shallow ocean going across the shelf, the continental shelf. And then beyond that, we have the bathyal zone. This is the deep water on the slope of the continental shelf. And then we have the abyssal zone. This is the deepest parts of the ocean floor. Now, organisms, these environments are very different, and there's very different organisms that live within each one of these marine ecosystem areas. So abyssal organisms are very different than you'll see in bathyal organisms as well as littoral organisms. So organisms that live in each one of these environments. They're very different environments. All right, so let's talk a little bit about some terms that we use for organisms in describing where they live in the marine system. This is a starfish and some wonderful brittle stars, some really cool echinoderms. And these are all living on the surface of the ocean floor, so we call them epifaunal. So these live on the substratum. We can have organisms that live in the substratum, that is, that they burrow down or dig deep. So those we refer to as infaunal. So these are organisms that burrow down or live within the substratum. And here's an example of a sand dollar, which is a echinoderm we'll talk about later, but is burrowing down where they graze. Here's a little movie of a sand dollar going along, burrowing in the sand along a beach. And the way that they make their living, their niche, is to go along and basically filter through as much of the sand as possible as they go along in the sand and filter out nutrients and food from the sand as they burrow in. So it's a really good example of an infaunal organism. As we mentioned before, we have benthic organisms. These are organisms that are living on the bottom of the ocean. So to be a benthic organism, you have to be some sort of constrained on the organism. And here's a scleractinian coral from Hawaii that's exhibiting a benthic lifestyle. But there's lots of ways you can be benthic as long as you're living on the bottom of the sea floor. Vagile is a different type of benthic organism. These are organisms that can move about. This is a photo from Antarctica showing a bunch of vagile organisms that are benthic. They're living on the seafloor, but they all can move around. Many of these are scallops, Antarctic scallops, and they basically can hop around and move around on the seafloor, as well as some echinoderms in there that can move around on the ocean floor, although they're not up swimming about like other organisms. Sessile organisms are benthic organisms, but they're tied down, constrained down to the ocean floor, so they cannot move about. This will include many of the corals and many of the organisms that exhibit that type of lifestyle. They also include many of the crinids that we'll talk about, such as the sea lily that you see here. So they're tied down to the seafloor, living on the seafloor. We refer to those as sessile. Pelagic organisms, on the other hand, are ones that live in the water column. Pelagic often means they're floating around in the water column. They don't necessarily have to be swimming, they just are floaters. And this is an example of a cnidarian. Nektonic is organisms that can swim, that can actively propel themselves around in the water. Here's a lionfish, and fish are a great example, as well as dolphins and whales, and any organism that really swims or actively swims in the water column. We refer to those as Nektonic. Planktonic are organisms that float around in the water. they may have very limited ways in which they can basically swim around. So this is an example of a jellyfish where they are just floating, kind of free floating in the water column. They have very limited control on the direction that they're moving around. They're just floating in and existing in the water column. We call that planktonic condition. So they're just floaters. Epibots are probably the coolest things. These are organisms that lived attached to other organisms, and we'll be talking a lot about epibots later on. So these are some examples of some shells of Cryptella on a horseshoe crab. And so there's a lot of organisms that will live on other organisms, and we'll be exploring a lot of organisms that make their living by just hitching a ride with some other organism. And that way they can be sessile, but they can also move around as well, as we'll talk about later on. So there's lots of really good organisms of epibots. So I want to talk a little bit about sort of amazing fossils where we can sort of reconstruct their life habits as a fossil. And this will give us some greater insight in how they are making their living. And then later on in the next video, in Lecture 10, we'll talk a little bit more about how we can use the fossil record to look at perturbations in the fossil record and recovery intervals, which are really key to understand paleoecology. This is Heracostrea, which is a brachiopod. Now, most brachiopods that you've seen before, just have those two shells. They're almost like clams, but they're a little different. But this is totally not like any brachiopod you ever seen. This is from the Permian of Texas, from the Guadalupan. And these guys have these like little open flaps there, and then they have this like weird, strange lower hinge on their bodies, and they're all different shapes. Brachiopods today tend to be very constrained in terms of their sizes and stuff. And so these guys were brachiopods that decided they wanted to be sessile corals. They wanted to be planted firmly down onto the substrate, and they form reef complexes during the Permian. And they're really fascinating. And one of the things that's really interesting is that many paleontologists debated how they actually opened on that hinge, because it's a very weak hinge. And we'll talk about brachiopods later on, but they have a very specialized hinge to open up and close. And so there's this huge debate of whether they opened their hinges or not, and how they actually would feed or live in these reef deposits in the Permian. And there was a fossil that was found where a brachiopod, another species of brachiopod, got in and was basically living within on top of there. And they realized something, that this hinge was non-functional. So what these guys would do is they would have their lophophores, which are the parts that they bring out to feed, that would sit in there, and they wouldn't open up the hinges. They would just keep it open, and that's how they would actually feed. But they were using that top hinge just sort of to protect them from predation. And so they didn't necessarily have to close it or open it like modern brachiopods and coeval brachiopods, like this one that found itself into this little net that lives there. So this is kind of a really cool example of an organism that just completely changed the habitat and niche that it fit into. They're really, really cool and fascinating, weird brachiopods. We'll be talking about some bivalves that do this as well later on, where they completely become sessile and more of a coral-like creature than the more mobile versions that they had earlier on, or the more normal version, I should say. Another really good example of a sessile organism becoming planktonic is with this crazy weird crinoid called Serracrinus. Now, Serracrinus is found in Europe and in the Jurassic, and they're just weird. They're basically like your typical crinoid. These are the sea lilies, and they, traditionally crinoids are going to be implanted on the seafloor. They're going to have a flowering part that comes up. This is the calyx that they can feed with their tentacle like arms. But what these guys did is they hitched a ride on things that float, particularly wood fragments. So logs and things would be floating, and these guys would attach to them and then dangle below the floating logs or organic material that would float. And so they would drift along the ocean dangling upside down and feeding in the water column. So this is a way of a sessile organism becoming mobile and being able to move around the landscape. And there's some other wonderful examples we'll be talking about of sessile organisms that became more mobile as they evolved through time. So another example of how strange and weird some of these habitats can be. So some organisms like crinoids that typically are going to be sessile and tied down to the ocean floor can suddenly find a way in which they can become more mobile and move around within the ecosystem and change their niche and change their habitat to do something new and unique. So one of the things you also want to look for is not necessarily just fossils, but also trace fossils or evidence of biological activity. That's going to be very key to understanding reconstructing ancient marine environments. So trace fossils, we'll be spending more time on trace fossils later on this semester, are really important. So these are marks that are left behind of grazing, of snail grazing. So these are gastropod marks or grooves that existed on this muddy beach where the snails went across and just were feeding and grazing as they moved across the landscape. So they left behind these grazing traces. Now you may not necessarily capture these gastropods. Maybe these particular gastropods don't have shales or hard parts, but you get the indication that they're there from the trace fossils. So trace fossils are very important to bring forth whenever you're doing paleoecological studies. All right, now we're going to talk a little bit about limiting factors. So these are some of the things that we can use the fossil record to try to get at in ancient environments. Things like temperature or climate, the amount of oxygen in the water depth, the salinity of the ocean, the substratum, and also something about the food or the food pyramid, food availability or nutrition that might be available to the organisms living at that time. So when we're reconstructing the environment, these are things that we can actually use the fossil record to use to reconstruct what the ancient marine system was like. So let's look at these real quickly and run through them. So temperature varies quite a bit in the world's oceans. Sea surface temperatures are the warmest around the equator, and as you move towards the poles, those sea surface temperatures drop off and become more cold. But the other thing to remember is that as you go deeper in the ocean, temperatures also drop and become colder. So the coldest ocean waters are found near the poles, and at the deepest part, the abyssal plains, the very abyssal deepest parts of the ocean, and those are going to be the coldest. And this is going to be important when we're reconstructing sort of climate through time, is knowing where you are in the water column. This pattern of warm surface water going down to cold water is called the thermocline when we look at it in the water column through the section. Another thing that's really important is oxygen. So oxygen is needed for many animals that live in the ocean, whereas many of the photosynthesizing organisms don't require oxygen. Now oxygen can be depleted, and when oxygen is depleted in the ocean, we refer to these as anoxic conditions. Now, anoxic conditions happen in many parts of the ocean today. A lot of times they happen along the coast of Louisiana, along the Mississippi Delta. This is caused by organisms, photosynthesizing organisms that bloom, like a lot of the algae, and they become extremely common, and then they die and they sink down. And organisms start to feed, scavenging organisms, on those algae blooms, and they use up all the oxygen. So there's no more oxygen for many organisms like fish, crustaceans, and this is a big issue with many fisheries and shrimp farms and stuff along Louisiana, is when you have these anoxic conditions, it means that all those organisms die. They just don't have enough oxygen in the water to live. And anoxic periods have happened in the Earth's path. There's a whole bunch of anoxic water during the Cretaceous periods, and these are periods where you get a lot of organisms dying out, and they're also characterized by changes in the water chemistry as well. So anoxic events are something to be aware of because they can have a pretty profound effect on the ecosystems. Water depth is one of the ones that I think paleontology has the best way of getting at water depth. And that's because organisms are really dependent on how deep they are. So the photic zone is really important because the photic zone is how far light will travel down. So many organisms have to live within the photic zone. They need that light to live and exist. So as you go deeper in the ocean, you have to, if you're an organism, you have to change the way in which you make your living, and you have to figure out a way to live in the dark, basically, if you're going to be down in the benthic zone of the aphotic zone, so the deeper, darker waters. The other thing about water depth is as you go down, temperature changes. You have to worry about salinity. So the saltier waters are near the surface, so there's greater evaporation, and deeper waters tend to be a little bit more fresh, but there's like what's called a halocline in determining salinity. And also density. So the water near the surface is less dense than the water at deeper depths, and that has to relate to the changes in salinity and the changes in temperature that reflect the differences in density. Now we'll be talking about corals and the importance of water depth to corals, but there are many corals that rely on a symbiotic relationship with photosynthesizing bacteria. These corals are called the hermatypic corals. Hermatypic corals are the ones that contain this thing called these bacteria called zoanthia. Zoanthia are photosynthesizing bacteria that live within the coral, and they need to be in the photic zone for that bacteria to survive. So the coral itself feeds on these bacteria, and the bacteria basically gets its energy from the sun in the photic zone. So if you ever hear of coral bleaching, we'll talk about that later on, that's when the coral loses its zoanthia, and then it basically dies of starvation because there's none of this photosynthesizing bacteria to feed on, and so that causes the corals to die. Ahermatypic corals are corals that do not contain zoanthia, and these are at deeper depths. So these are corals that live at very deep depths, and they don't have the bacteria that require photosynthesis. Salinity varies quite a bit across the oceans. There's certain parts of the ocean where salinity is very high, and there's certain parts of the ocean where salinity is very low. Salinity tends to be highest where there's the greatest amount of evaporation. This includes like the Red Sea, the Mediterranean, as well as some of these open parts of the Atlantic Ocean in the middle of the ocean gyre. So these are areas in which there is significant evaporation going on. Places where the ocean water tends to be more fresh is in Southeast Asia, where you get a lot of runoff of fresh water from these monsoonal conditions that can happen across India and Southeast Asia, as well as in polar areas where you have a lot of glacial snow melt and sea ice melt that brings in lots of fresh water seasonally into the oceans. So salinity is a major factor. Now, organisms can either have a great tolerance in salinity, or they can have a very narrow tolerance in salinity. So stenohaline organisms have a very narrow range of salinity, whereas euryhaline organisms are organisms that can live in a very wide range of salinity. So to give you some salinity numbers out there, so ocean salinity is between about 30 to 40 parts per thousand. Brackish water is about 0.5 to 30 parts per thousand. So brackish water is going to be where you're going to find fresh water coming out into the ocean. So these are estuary type environments. Fresh water is where salinity is below 0.5 parts per thousand. And then hypersaline are where you get like 40 to 80 parts per thousand. So very, very salty places. This is hypersaline you get like in the Great Salt Lake here in the United States. And then you get brine. Brine is above 80 parts per thousand. So these are very salty places like the Dead Sea in Israel. Some places like that where it's very salty. Now, many organisms can live in certain salinity levels, but brine conditions are probably the most severe. Only just a handful of organisms can live in brine water. So salinity is very important in terms of where organisms can live. And so knowing those tolerances and the limiting factors in the fossil record will help you reconstruct the salinity in the ocean. Another really important one that we usually don't consider but is very important for the organisms that are living in the marine ecosystems is the substratium, that is the surface that they are living within. So you can imagine mud, a mud substratium. Living in mud is going to be very different than living in sand or gravel. So if you're along a gravel beach or sand beach, it's going to be a very different environment than if you're living in a muddy sort of tidal flat. And so organisms tend to live in different environments based on the substratium. So many mollusks will burrow down. We'll talk more about this when we talk about them in more depth, but they'll burrow down the substratium to live in sort of muddy areas, and they need a certain amount of, if it's too muddy or too mucky, then they may have trouble living in that type of habitat. Whereas if it's more sandy, they might find it more beneficial. So imagine sand dollars needing that sand, and so the substratium is pretty important for their feeding and the way that they make their living, their niche. Some organisms like mussels, of course, can burrow into hard rocks and stuff, and they don't necessarily need mud. They actually need like a rocky surface where they can attach to and grow from, and so that becomes their substratium. So we'll talk more about substratium when we talk about these various mollusk groups and the way that they attach to the substrate, but these are very important things to consider when you're reconstructing this ancient marine environment. So food is another limiting factor that we should consider. So understanding each of the fossil organisms' role within how they actually get their food is going to give you a greater insight into reconstructing this ancient marine environment. So you can have organisms like starfish that are carnivores, and that way they're going to be feeding on other organisms, or you can have organisms like this gastropod, this little snail that's a grazer, and so these are very different organisms, and you might find them in very different environments, and so finding them is going to give you a greater insight of what the ancient marine environment was like. So hopefully this video kind of enables you to think about some of the things that paleontology can bring forth when you're reconstructing the ancient marine environment. All right, next time in Lecture 10, we're going to talk a little bit more about cyclic perturbations that can happen in the marine system and how communities can recover and change, and how we might be able to see that in the fossil record. So thanks for watching. If you're interested in taking a course in geology at Utah State University, you should check out the department website at geology.usu.edu. And if you're interested in who I am and my own research, you should check out my website at benjamin-berger.org.